Hello students, welcome to Distinction Learning Platform, and on this platform you find revisions, tests, quizzes, a lot of stuff. So before we begin our lessons, make sure that you subscribe so that you can be updated for upcoming videos. To make the story short, let us begin. In this video, we're talking about glycolysis, and this is under what we call biochemistry. Okay, so you need to pay attention when it comes to glycolysis because there are a lot of things and important things that we'll be talking about. Okay, so you need to pay attention. So what is this same glycolysis that we're talking about? So glycolysis basically is a metabolic process or biochemical process that's going to be using glucose in order to produce ATP, and mostly it's going to occur in the what or cytoplasm. Okay, so what are we saying about glycolysis? So we are saying glycolysis is going to what? It's a metabolic process. So we are saying metabolic process, okay, process that uses what? That uses glucose to produce what? ATP. Okay, so it's going to use glucose to produce ATP. So we'll talk about this glycolysis because there are a lot of processes that are going to use glucose in order to produce what? ATP. Okay, and make sure that you need to understand about the glycolysis is that there are about what? 10 processes that are going to involve enzymes. So they are enzyme catalyzed type of what? Processes. So under glycolysis, we're going to have about 10 steps. So we'll understand step by step. Okay. Now, why are we talking about glycolysis? Okay, so glycolysis is a very important thing. When you talk about the red blood cell doesn't contain mitochondria. Okay, so if it doesn't contain mitochondria, what does it get ATP or energy? It gets from what? The glycolysis. Why? Because glycolysis happens in the cytoplasm. Red blood cell doesn't contain mitochondria. What else apart from that? Also, when someone is doing what? Some exercise, muscles, they need what? ATP. Okay, so rapid production of ATP is done by what? Glycolysis. Instead of going into the electron transport chain to produce a lot of ATP, but it will depend on the what? Glycolysis. Why? Because glycolysis is a fast type of what? Metabolic process. Then apart from like someone who is doing some exercise, what else is glycolysis important? Remember that also in the brain. In the brain, glycolysis is important because that's a source of ATP for the normal function of the what? Brain. Okay, so you need to understand about that. So that's why glycolysis is very important. Okay, now let's proceed. Let's talk about something important. Okay, so let's talk about the key facts about glycolysis. What are the important things that you need to know about glycolysis? So what are those? So the first important thing that you need to know is what? The location. Okay, so this is what we call location. So what did I say about the location? I said to say location glycolysis, it can happen in the what? Cytoplasm of all cells. All cells, glycolysis is going to happen in the what? Cytoplasm. So the key important thing that we need to understand here under location is going to happen in the what? Cytoplasm of all cells. Okay, that's the most important fact. What is the other fact that you need to understand? Oxygen. Okay, does this glycolysis uses oxygen in order for energy to be produced? No. Okay, so here we are saying glycolysis is independent of what? Oxygen. So you need to understand about that. Okay, doesn't use oxygen. Okay, then what else? What is the starting molecule? So here, what is the key fact about the starting molecule? What is the molecule that's going to be start with? It is what? Glucose. So the starting molecule is what we call what? Glucose. For glycolysis to happen, there has to be what? Glucose. If there is no glucose, glycolysis can't happen in the cytoplasm of all cells. Okay, so what else? What is the now here? This is what we call final product. So what will be the final product? So the final product, I'm not talking about ATP. We know that ATP is going to be produced, but what could be the final product for you to say that glycolysis has ended? What is that final product? That is that molecule is what we call pyruvate. Okay, we call it what? Pyruvate. Okay, pyruvate. So that is a 3-carbon molecule. So here is a 3-carbon organic molecule, and we know that glucose is what? The 6-carbon molecule. So as you can see, the 6-carbon molecule is converted into what? The 3-carbon molecule. So very important to know about that, okay? So glycolysis is going to use what? The 6-carbon molecule, which we call glucose, and break it down into what? The 3-carbon molecule, which we call pyruvate. Pyruvate is the final product of glycolysis. Okay, so now if you come here, what is this? This is what we call net ATP. So net ATP, it means that it is the final ATP that you are going to have after what? The summation of other ATP, because there will be ATP that we are going to put into certain reaction that we'll be talking about, and the ATP that we are going to get. If you find the sum of those, then you're going to have what? Net ATP. So what will be the net ATP? The net ATP of the glycolysis pathway is what? 2. So we are going to have what? 2 ATP. 2 ATP means that the one that we are putting in and the one that you gain from, all together you're having about what? 2 ATP. Okay. Then what about the net NADH? So NADH, nicotinamide adenosine diphosphate. So what do we call that? So how many are these molecules? We are going to have about what? Also 2, about what? 2 NADH. Okay. NADH is also very, very important in what? The glycolysis pathway. Okay, it's very, very important to talk about it, because something that's going to carry electrons. Okay, it's very, very important one. So these are the important facts that you need to understand. What else apart from that? The other important fact that you need to understand is that, what did we say? We said if you get glucose from this equation, we're saying, let's just say G, if you get glucose, okay, then you're going to, what are you going to get? You're going to get what? Pyruvate, okay, pyruvate plus what? ATP, okay, but not only ATP. Not only ATP. So let's get something here. Let's try to understand something here. But not only ATP. Let me use the blue. Okay, so we are saying glucose, one glucose molecule. Let's just say one glucose molecule. Okay, it's going to produce about how many? 2 pyruvate molecules. So let's just say P. P stands for what? Pyruvate. So from each glucose, since remember that, let's just do simple mathematics here. Okay, so we said this is a what? A 6-carbon molecule, which is what? Glucose. So we are breaking down the 6-carbon molecule into what? Into what? 3 carbon molecules. Okay, so meaning that we're going to have a carbon molecule with what? 3. Another carbon molecule with what? 3. Okay. Okay, so that we are going to have 3 carbon molecule, another 3 carbon molecule, so that all together we are going to have what? 6. And what do we call this 3 carbon molecule? We say that is what? Pyruvate. No wonder we are going to have how many pyruvate molecule? 1 pyruvate molecule and another pyruvate molecule. Why? Because we are breaking down large or 6 carbon molecule into what is smaller 3 carbon what molecules. And how many are they? 2. So now we are going to have what? 2 pyruvate molecule. So what else apart from that? We're also going to have, remember that we're going to have what's the net ATP here? We're going to have what? 2 ATP. Okay? And what else? What else? Remember that we had also NADH, which is the electron carrier molecule. Okay, we're also going to have what? 2 NDH. Okay? This one. So we're going to have all those. So you need to understand about this net equation, okay? So this is a very, very important one if you need to understand about that, okay? So this is the main thing that we'll be talking about. Now, since we've understood the introductory part of what? Glycolysis, what else are we supposed to understand? We need to understand about what? The phases. So since we remember that glycolysis is going to have about how many steps? 10 steps. So to make things simple, they have divided the 10 steps into what? Into 2 main categories. The first 5 steps and the last 5 steps. So they are going to have what? Each group. So the first group and also the second group. So which one is the first group? So the first group is called what? This one, which is the preparatory phase, okay? This one. And the last group is going to have harvesting what? Phase. So you are going to find out to say preparatory phase is going to have what? Another name. So the other name for preparatory phase is what? The energy investment what? Phase. So if you see something like that, same as the preparatory phase. Harvesting phase, so the other word that they can use is what? Energy payoff phase. So you need to understand all those terms that they like to use. Okay, so we're going to start with this one. But before we start, we begin the phases. There's an important thing that you need to understand because it's a lot. Okay, so you need to be familiar with these words. Okay, these words from 1 up to 10. If you are familiar with this, it will be very simple to remember all the enzymes and everything that we'll be talking about. Okay, it will be very easy. So remember that we're starting with what? Glucose, and we're going to end with what? Pyruvate. Okay, we're going to end with the pyruvate. Now let us look at the names of these molecules that are between the glucose and what? Pyruvate. What are those? The first one is what? This one. Number 1 is glucose 6-phosphate. That is the first one. The second one is what? Fructose 6-phosphate. Okay. The third one is what? Fructose 1,6-bisphosphate. The fourth one is what? Glycolysis 3-phosphate. And the fifth one is what? Dihydroxyacetone phosphate. So these are the first 5 important molecules that are important in what? Phase 1, or the preparatory phase, or what? The energy investment phase. This is the one that I'm talking about. So from 1 up to 5, they will be under what? The first phase, okay, the preparatory phase. Then what is the last one now? The last one is this one. The harvesting phase is going to contain what? It's going to contain these molecules 1,3-bisphosphoglycerate. Another one, number 7, it is going to contain 3-phosphoglycerate. Then 8 is going to have 2-phosphoglycerate. Then number 9, we are going to have phosphoenolpyruvate. And number 10, we are going to have what? Pyruvate now, which is the final product. So you need to be familiar with this. So for you to proceed, make sure that you are familiar with what? These names, or maybe you can pause the video so that now you can look at them. If you are familiar with that, then we can proceed with what? The first phase. Very, very important, okay? So now, since we are familiar with this, now we can go to the what? To the phases. So before we go to the phases, what is the main important thing that you need to understand? How many phases that are there? Okay, how many steps that are there? We said, so under phases we're going to say there are about how many steps? There are about 10, let's just say, steps characterized by what? Characterized by enzymes. So these steps, these 10 steps, they are not just going to what? To happen on their own. There has to be what? An enzyme. So each step is very important because it's going to be characterized by a certain or specific what? Enzyme. So you need to be understanding about that. So basically, if you talk about glycolysis, to be more specific, you are saying, okay, it's 10-step enzymatic type of what? Metabolic process that's going to what? Use glucose and produce what? Pyruvate molecule, and in due to that, we're going to have what? About 2 ATP or 2 net ATP production, and that is what we call energy production. Okay, so you need to understand about that. Okay, now let's begin with what? The preparatory phase. So let's start with the preparatory phase. So preparatory phase, what's the other term that they use? So the other term that they use is this one. We said energy investment phase. Okay, energy investment phase. Why? Because we're going to put what? ATP. So the reason why it is called energy investment phase, because we're going to put in these processes, then the amount of ATP that we're going to put is about what? 2. So we're going to put 2 ATP in this process, okay? 2 ATP is going to be used up. So 2 ATP in the preparatory or investment phase is, so the 2 ATP is going to be what? Used. Okay, so you need to take note of that. Now, what's the first step? Okay, so now the first step is this, so you need to pay attention now. Okay, so the first step is like we're going to get what? Glucose. Remember that we need to understand where glucose is coming from. Maybe someone during the phase state, someone was eating or feeding like that. Okay, glucose will find itself on what? In the blood. Okay, so let's try to imagine to say this is the glucose that came from different systems like that. Okay, so let's say this is our glucose, so let's use black. So this is what? Our glucose, which is within the what? The circulation. Okay, that is glucose. Now for it to enter into this, so what do we call this? So let's speak, this is what? A cell. So this is a certain cell. Okay, this is a certain cell. Now this cell, we can pick which one is a common cell that we're going to use. We can use red blood cell, we can use hepatocytes. So let's just say it's a liver cell. Okay, so let's pick this is a liver cell. Okay, this is hepatocyte. Okay, now these glucose molecules, they want to enter into what? Hepatocyte. Okay, now they don't just diffuse into the hepatocyte, they need to pass through what? Certain transporters. Okay, on this hepatocyte or liver cell, they are what we call what? Specific glucose transporters. Okay, so on the cell membrane of the what? Hepatocyte or the liver cells, there are some transporters for the what? For the glucose. Okay, and if you look at this blue, if you look at that blue transporter, how can you call that? So we call it what? A glucose transporter. So it's just a simple name, glucose transporter. Now we abbreviate this as what? We abbreviate this one as what? GLUT. Okay, GLUT. Okay, so we're going to abbreviate that one as GLUT, glucose transporter. Okay, so glucose transporter. So now there are different types of GLUT transporters. So GLUT, we have GLUT 1, we have GLUT 2, we have GLUT 4, we have GLUT 5 as well. Okay, but the most important GLUT transporter that can be found in the what? In the liver are what? That is GLUT 2. So GLUT 2 can be found mostly in the liver. Okay, but what about the red blood cells? The red blood cells are going to have GLUT 1. As well as what? The neurons, they are going to have GLUT 1 as well as what? GLUT 3. GLUT 3 can be found in the what? In the neurons as well as what? In the red blood cells. Okay, then what about GLUT 4? Where can you find GLUT 4? GLUT 4 or glucose transporter type 4, they can be found in the what? Basically you can find them in the in the muscle cells there. Okay, in the muscle cells, adipose cells there. That's why you can find what? The GLUT 4. And GLUT 4, they are regulated by what we call the insulin. So those are the important things that you need to understand. But here, let's just try to understand to say, this is what? The GLUT transporter, where glucose is going to what? To pass through. So this glucose, as you can see, this glucose that is coming in, so let us use black. So this glucose that is coming here, so it's going to diffuse here. Okay, it's going to enter into this hepatocyte via that GLUT transporter. Remember that in the liver, what type of GLUT transporters that are there? So basically, you're going to have what? GLUT 2. Okay, so I'm going to have GLUT 2 transporters. So glucose is going to diffuse like that. Okay, it's going to diffuse down the concentration gradient. Why? Because in the blood there is a lot of glucose here compared to the what? Hepatocyte. So it's going down the concentration gradient. So that's how glucose is going to what? Diffuse. So let's say now inside here we're going to start using blue. So this is glucose which is inside. Okay, now you can ask yourself to say, this glucose that which is within the hepatocyte, it can go back. Okay, because it's just diffusing via the what? The GLUT transporters. So basically this glucose will go back into the bloodstream if nothing is going to happen to that. So what do you think is going to happen to this glucose? So for you to prevent this glucose from leaving this cell, because glycolysis has to happen in this cell. So what do you think? We're going to put a phosphate onto this what? Glucose. What do we call that? Phosphorylation. So for you to prevent glucose from escaping from the what? Hepatocyte back into the bloodstream, we're going to put a phosphate what? To that glucose. Okay, that's what we call phosphorylation, and that's the first step of what? Glycolysis. So this glucose which is here, so we're going to put here glucose. So this is the glucose that we have within the what? The hepatocyte. So to prevent this glucose from leaving this cell, we're going to put a phosphate to what? To this glucose. Okay, we're going to put a phosphate to this glucose. If you put a phosphate to this glucose, then we're going to say, now it depends on the what? On the number of carbon one phosphate is going to be added. But remember that glucose is a what? 6 carbon molecule, but the phosphate is going to be added where? On the 63 carbon. No wonder this molecule that we're going to call here, we're going to call it glucose 6 phosphate, because the phosphate that was added, it was added on what is 63 position of carbon. Okay, so we're going to say the product that we're going to have here is glucose what? 6 phosphate. So I'm going to say G6P. Okay, glucose 6 phosphate. Why? Because this 6 basically it means that this phosphate was added on the what? 60th carbon of what? Glucose. Now, the question that we can ask is that, okay, quite alright, this glucose can't leave the cell. Why? Because this is now, it has a phosphate and it is negatively charged. If it is negatively charged, it can't move out of the what? Of the cell. Okay? It can't. Now, the important question that we can ask is that, where is this phosphate coming from? Where is this phosphate coming from? It has to come from where? From something else, okay? It has to come from here. So, we are going to add ATP. So, ATP, adenosine triphosphate, is going to have a phosphate. So, from ATP, we are going to get a phosphate from ATP so that we can put to glucose, okay? So, we are saying from here, we are going to put what? Okay, we're going to put ATP. We're going to put ATP in what? In this glucose, so that the glucose can obtain this phosphate. Okay, remember that now if you get ATP, if you get now ATP, if you get one phosphate from ATP, then what are you going to have? You're going to have ADP. So you're going to have what? ADP, adenosine diphosphate, because you had 3 phosphate and you used 1 phosphate to get what? Glucose 6 phosphate. Now you're going to have di, means you're going to have 2 phosphate. Yeah. So this is the thing that's going to happen here. Okay. So we're going to remove the ADP there. Now the other important thing is that there has to be the enzyme that has to regulate this process, because this is not happening on its own. There has to be the enzyme that's going to convert glucose into glucose 6 phosphate. The one that's going to use ATP. What do we call that? Okay. Basically, there are particular 2 enzymes that are important here. Okay. What are those? So the first enzyme that's going to catalyze this reaction, what is that? We call it hexokinase. Okay, so you need to know this one. Hexokinase can be found in all the cells. Okay, all the cells you can find hexokinase. Okay, hexokinase is the one that's going to get glucose and convert glucose into what? Glucose 6 phosphate by using ATP. Okay, but there is also another enzyme that can do the same thing, which can also be found in the particular what? In the particular organ. What do we call that? Glucokinase. So you need to understand something here. So glucokinase can also do the same thing, can convert glucose into glucose 6 phosphate by using ATP as well. Now the major difference is that glucokinase can be found in the what? Liver, as well as what? Pancreas. So let's just say pan. Okay, so glucokinase, it is limited in the what? Liver and pancreas. Okay, so since this is the liver we're talking about, we are going to use what? Glucokinase. So glucokinase is the one that is converting glucose into glucose 6 phosphate. Okay, now this step, basically this step is what? Is not the forward and reverse what type of reaction. It is just what? The forward reaction. It is irreversible. Okay, it is forward. Okay, now the important thing that you need to understand about this step is that there has to be some things that are going to inhibit this what? Conversion. Okay, what are those that are going to inhibit this conversion? Okay, what are those? Basically, if you have a lot of ATP, it's going to prevent this conversion. Okay, so we'll talk about the substances that are inhibiting the certain conversion. Okay, because this conversion is a very, very important conversion under glycolysis. Okay. Okay, now let's proceed to this. Let's proceed. Now we're going to get this glucose. Okay, so let's proceed to another thing. Okay, another step. So another step now, we're going to get this glucose 6-phosphate and convert it into something which is very, very important. We're going to convert it into what we call, okay, fructose 6-phosphate. If you follow this one, we said the first reaction was with glucose 6-phosphate. Now the second one is what? Fructose 6-phosphate. How does that happen? Okay, remember that fructose is a what? 6-carbon molecule. What about glucose? Glucose is the what? 6-carbon molecule. Okay, so if these are two like type of what? 6-carbon molecule, there is nothing that is changing. We are not adding any phosphate in this what type of reaction, but we're just changing, okay, the structure. Okay, so remember that if you look at glucose is a what? Is the 6-sided type of what? Ring. Okay, the type of a ring for glucose, okay, is like this. Okay, it is what? It has 6 sides. 1, 2, 3, 4, 5, 6. Okay, it's going to have what? It's a 6-sided type of what? Ring. Okay, this is glucose. Now if you look at fructose is more like the 5. It looks like that. As you can see, how many sides are there? There are about what? 5 sides. Okay, so basically what is going to happen here is that we're changing just the ring, okay, from the 6-sided ring to the what? 5-sided ring. Okay, basically what is happening here is what? Isomerization. Okay, just basically it's an isomer what we want. So we want an isomer from what? Glucose 6-phosphate. Okay, so what is really happening here is that's what? Isomerization by a certain enzyme. Now what type of enzyme is going to help this isomerization? What type of enzyme? Very, very simple. So this enzyme here is what we call, so let us write it in what? In red. So the enzyme that's going to help to convert this glucose 6-phosphate into fructose 6-phosphate is what? Okay, so we call it what? Phosphoglucose isomerase. Very, very important. As you can see, I'm saying isomerase because this is isomerization. Okay, isomerization. So it is phosphoglucose isomerase. Very, very important enzyme. Okay, here there is nothing that's going to happen, yes, because this is a reversible reaction. Okay, this fructose 6-phosphate can be converted back to glucose 6-phosphate. It's not a very, very important step. Okay, next we go to another one. Let's go to this. Okay, let's go to this. Now to this, remember that fructose 6-phosphate, when I said this is a very, very important what table, we're going to have this one. Okay, we're going to have fructose 1,6-bisphosphate. Okay, so we're going to have this. I'm going to explain on this one. We're going to have what? Fructose 1,6, okay, bisphosphate. Okay, let me just write it in full. So fructose 1,6-bisphosphate, okay, sometimes abbreviated as what? F, okay, B, P, okay. So let's just abbreviate it as 1,6 like that, okay. So let me just write it well, okay. So it is abbreviated as what? So let me just use black. So here it's abbreviated as what? Okay, F, like that. Okay, F. Okay, then you are going to put 1,6B like that. Okay, so it's going to be abbreviated like that. Fructose 1,6-bisphosphate. Okay, that is what we call what? The product where fructose is going to be converted on that. Now you need to understand something here. Okay, as you can see, we're having 1,6. So basically, on 60th position, what did we have? We had phosphate on 60th carbon, but again here we are putting another phosphate on the what? First carbon. And remember that what is the rule? If you are putting phosphate, basically you're going to add what? ATP. You need to understand about that, okay? So here we are going to add what? ATP, because we are having another phosphate on first carbon. If you're having another phosphate, the reason why B basically bisphosphate, we are having two phosphate in the what? In a certain what? Carbon molecule. Okay, this one. We're having about two phosphate. Now, why is another phosphate coming from? Because initially we had just one phosphate on 6 carbon. Why is it coming from? It is coming from what? ATP. It is coming from ATP. So we're putting ATP again in this reaction. So this ATP will be converted as usual into what? ADP. Okay, so we're going to get one phosphate from this ATP and convert it into what? ADP, like that. Okay, so that we can have fructose 1,6-bisphosphate. Bisphosphate means two phosphate. Okay, the phosphate on the first carbon and the phosphate on the what? 6 carbon. Okay, now another important question is, what is the enzyme that's going to catalyze this reaction? Because each step has to be catalyzed by what? A certain enzyme. So what is that enzyme that's going to catalyze this? Okay, so what is that enzyme that's going to catalyze this one? So this one is what we call, this one is called phosphofructokinase. Okay, one. Okay, so this is a longer type of enzyme. So we're going to have phosphofructokinase. So the moment you just say kinase, it's going to use what? Energy. Okay, make sure that you understand it, because if you come back here, we had exokinase or glucokinase. So anything that's going to end with kinase, basically is going to use ATP. As you can see, under here we're having ATP. But this step where phosphofructokinase 1 is going to convert phosphate fructose 6-phosphate into what? Fructose 1,6-bisphosphate, is a very, very limiting step. This step is very, very important. Okay, so here we're saying this step is what? We're going to call this step as what? A limiting step. Limiting step. What does it mean? It means that if these molecules doesn't reach, they don't pass through this what? This phase. Okay, this step where fructose 6-phosphate is converted into this molecule. If that doesn't happen, glycolysis is not going to occur. Okay, the complete glycolysis is not going to occur. But the moment this step happens, now the glucose has been activated so that now it can go to glycolysis pathway, known as a limiting what? Step. And it's just what? It's not a forward reverse reaction, but it's just a forward type of what? Step. Okay, it's a limiting one. And if you remember that since it is a limiting one, okay, it's going to be what? It's going to be inhibited, or this step is always, it doesn't like what? Okay, let's start with something that it likes. So what are the things that are going to promote this step? So you need to know, because you're going to be examined on this. So what are the things that are going to be what? Promoting this step? The first one is what? AMP. So AMP, okay, adenosine monophosphate, is going to promote this step. What is the other one? ADP. Okay, ADP is also going to promote what? This step. And the other important enzyme that's going to what? Promote this step is what? We call it what? We call it phospho. What do we call? It's another molecule. Sorry. So another molecule that is important as well. Instead of saying fructose 1,6-bisphosphate, it is what? Fructose. So the other important molecule is what? Fructose 2, okay, 6-bisphosphate. So fructose 2,6-bisphosphate is going to promote this reaction. Okay. Now, what is the things that are going to inhibit this what? Reaction? The first one is what? ATP. So a lot of ATP is going to inhibit what? This limiting step. What else? Also, this product that we have, fructose 1,6-bisphosphate, is going to inhibit the reaction. Okay, this one. So fructose now it is 1. So here make sure that you take note of this. So 1-bisphosphate is going to inhibit this step. 2,6-bisphosphate is going to promote. You need to understand about that. Okay, what else? Also, acidosis. Okay, acidosis, basically you have low pH, okay, less than 7. So then you're going to have acidosis, okay? So that acidosis or metabolic acidosis is going to what? It's going to inhibit this step. So a lot of factors that are going to inhibit this step. You need to know factors that promote this step and the factors that are going to what? Inhibit this step. So let's continue. So let's go to another one, okay? Another one now, as you can see, this fructose 1,6-bisphosphate, if you come here, fructose 1,6-bisphosphate is going to be what? Broken down, okay? It's going to be broken down into two important substances. What are those? The first one is what? This one, glycolyl 3-phosphate. So it's going to be broken down into what? This one, glycolyl what? 3-phosphate, okay? It's going to be broken down into glycolyl 3-phosphate. It's not glucose 3-phosphate, no. Glycolyl 3-phosphate. You can look at it here. Glycolyl 3-phosphate, okay? It's going to be broken down to that, okay? Now, by which enzyme? And also another one. So we said two. So because this is glycolyl 3-phosphate and another one which is what? Dihydroxyl acetone phosphate. This one, let's just call it DHAP. Okay? DHAP is what? Dihydroxyl acetone phosphate. So this is going to be broken down into these, okay? And this process also is going to require what? A certain enzyme, important enzyme, okay? The enzyme that's going to catalyze this reaction is called what? What do we call this? We call it aldolase, okay? So aldolase is the one that's going to convert fructose 1,6-bisphosphate into what? Glycolyl 3-phosphate and what? Dihydroxyl acetone phosphate, okay? Now, you need to understand something here. This D, so mean that if you look at this G3P or glycolyl 3-phosphate, this one is the one that's going to continue in the harvesting phase. But this one is useless, okay? It's not going to be used in the glycolysis pathway or in the next step, okay? So now the body is so clever to say it's going to get what? DHAP or dihydroxyl acetone phosphate and convert it into what? It's going to start converting this into what? The glycolyl 3-phosphate, okay? It's going to get this DHAP, convert it into what? G3P. Why? Because G3, let's just put here, okay? So what we're saying is here, we're saying, okay, this one is going to be converted into what? G3P, okay, will be converted into that because G3P is the one that's going to continue in the harvesting phase. As you can see now, how many G3P do we have here? We have two. There's this one that was initially produced and also this one that was what? That is now produced, okay? So no wonder if you look at glucose, one molecule of glucose are going to have what? Two products or two major products. The one that is what? Coming initially, and also the other one that's going to be produced what? Here, okay? So two type of what? 3 carbon molecules, okay? Now, here there has to be what? Also the enzyme that's going to convert dihydroxyl acetone phosphate into what? Glucose 3-phosphate. There has to be another enzyme. What do we call that? We call it what? Triose. So remember that tri stands for what? Tri stands for 3. So this is a 3 carbon molecule. Now, this triose, so this triose is what? This is a triose phosphate what? Isomerase. So this is what we call triose phosphate isomerase. So I'm going to say P here. We're going to use isomerase here. Okay? So we're going to say the enzyme that's going to convert DHAP into G3P is what? Triose phosphate isomerase. This is an isomerase because this is a 3 carbon molecule and also this is a 3 carbon molecule, okay? So no wonder this is just also isomerization. So that's where the first phase is going to end from, the preparatory phase, okay? Now let's go to the harvesting phase. So let us finish up. So our vesting phase now, remember that we are done with this from 1 to what? 5. We are done with that, okay? Now we can start with this one, okay? Now we are going to get this. We are going to get a G3P. So what is G3P? Glycolyl 3-phosphate, okay? Glycolyl 3-phosphate is going to be converted into something, okay? It's going to be converted into something. What is that something? This is 1,3-bisphosphoglycerate, okay? 1,3-bisphosphoglycerate. What is that? We're saying it's going to be converted into what? 1,3, let's just say bisphosphoglycerate. Let's just say 1,3-BPG. So what this is 1,3-bisphosphoglycerate, okay? It's going to be converted into that. Okay, now remember that there has to be the enzyme. The enzyme that's going to catalyze this reaction is what we call what? What we call that enzyme? We call it what? We call it G3P hydrogenase. Okay, the enzyme that's going to catalyze this conversion G3P into what? 1,3-bisphosphoglycerate is what? Glycolyl 3-phosphate hydrogenase. Okay, and this is what we call the oxidation phase. Okay, the oxidation reaction. Okay, so this is what we call oxidation reaction. Okay, so we need to understand about this oxidation what? Reaction. So this is the oxidation reaction. Now, for this one to happen, we are going to do something. So we're going to put, now instead of putting, we're not putting anything, we're not putting the ATP here, okay? Because the ATP, you need to put it in the what? Preparatory phase. As you can see, how many ATP did you use from the preparatory phase? We used this one, we used about what? 2. So we used about what? 2 ATP from what? Preparatory phase. Because this was the first ATP that we used, and also the last ATP that we use. How many? There are 2 ATP that we are going to what? To produce. Okay? So now here we're not going to use ATP, we're going to put something. For us, as you can see, 1, 3, we had just 3 phosphate. We had 3 phosphate on what? Third carbon here. Now we're having another phosphate. So now, where is that phosphate coming from? It's not coming from ATP. It is coming from what? The phosphate ion itself, okay? So here we're going to put what? We're going to feed the phosphate ion, okay? And also we're going to put what? This carrier molecule, electron carrier molecule, this one. We're going to put this one. NAD plus or NAD plus, okay? That one is also going to be what? Fed into this what? This reaction. Okay? If you put this one phosphate and NAD plus into this reaction, then we are going to also have, what is the other product that we are going to have? NADH. Very, very important. Okay, so we are also going to have that one, because as you can see, this NAD plus will go to the what? To the phosphate there, and it's going to get the what? The hydrogen from there. Okay, if it gets hydrogen from there, then basically it's going to form what? NADH. But again, since this is, you are going to have what? Hydrogen ion production as well. Okay, then we can continue. Since this is what is happening here, we can continue to another one. Okay, we're going to get this 1,3-BPG and convert it into what? We're going to convert it into what? The 3. Okay, so we're going to convert it into what? 3-phosphoglycerate. So let's just say 3G, like that. Okay, so we're going to convert it into what? 3-phosphoglycerate. Okay, again, so let us finish up. Okay, now remember that each reaction is going to contain what? A certain enzyme, okay? Each reaction is going to contain a certain enzyme. Now, what is the enzyme that's going to convert 1,3-bisphosphoglycerate into what? 3-phosphoglycerate. Okay, so the enzyme that's going to be used here, okay, it is what we call here the enzyme that's going to convert the 1,3-BPG is what we call phosphoglycerate, okay, phosphoglycerate kinase. Okay. Now remember the rule that we said is that since there is kinase here, so if there is kinase here, what do you think? You are thinking that we're going to put what? ATP there, okay? But we're going to put something so that we can produce ATP, okay? So we're going to put what? Here we're going to feed something which is important. We're going to put what? ADP here. So we're going to put ADP there, okay? If you put ADP here, then you're going to produce what? ATP. So very, very important. This step where you're getting this one, this molecule, and converting into 3-phosphoglycerate, we're going to use phosphoglycerate kinase. And this kinase enzyme is going to use ADP and convert it into what? ATP. Okay. Now, another important question, okay, or important concept that you need to understand is that this ATP, how many ATP are going to be produced here? There are two. Although you can see one, but there is also another reaction. Remember that how many G3 that we have here? We have this one, and also we have this one. Basically, let's say this one, this reaction, the G3P, it is the first G3P, okay? Then G3P is the second one. Also, the same thing is happening from the what? Other G3P, okay? Also, if that one is also happening, it will reach also the same step, okay? This also is going to occur. Remember that if this is ATP coming from the first G3P, and also the other ATP will come from what? Another G3P, okay? So basically, how many ATP are you going to have here? You're going to have about what? 2 ATP. And if you come back to this NADH, how many are we going to have? Also, we're going to have what? 2 NADH, okay? You need to understand about that. Very, very important number. So those numbers are going to be what? Asked. Okay, so now we can proceed. Now this 3PG, okay, is going to be converted into something. What is that? Okay, so it's going to be converted into this one. Okay, if you look at this one, we have this one. It's going to be converted into this. Okay, it's going to be converted into 2PG. This one. It's going to be converted into what? 2PG. Remember that 2PG is basically, it means that it is what? 2-phosphoglycerate. And what is happening here? As you can see, the phosphate here was on what? Carbon 3. But it has been moved to what? Carbon 2. What was our rule? The rule was that if you move a phosphate, okay, or maybe you are changing a ring. Remember that here, isomerization here, what was really happening here? On this one, if you remember, we said to say it's isomerization here because we are changing the ring, the 6 carbon ring into what? Another ring, the 5 carbon ring, okay? That was isomerization. If you also look at here, the triose phosphate isomerase, we are changing also the phosphate which was on a certain position and they put it into what? Another position. Also here, this is what is happening. This is isomerization. No wonder we're going to get a certain name here. So here, what is the name of this enzyme that's going to catalyze this reaction? It is what? Phosphoglycerate. So let's just say phosphoglycerate. Okay. Now instead of saying isomerase, we're not going to use that. We're going to use what? This one. So the correct name is what? We're going to put what? Mutase. So don't get confused on that. Okay. It's a mutase that's going to do that. Okay. So now what is the other thing? Let's get now this 2PG and convert it into what? Something, which is this one. Phosphoinopyruvate. This one. So let's just say PEP. Okay. Phosphoinopyruvate. So because it's a bigger name like that. Okay, it's longer. So let's just call it PEP. So we're going to get what? 2PG and convert it into what? PEP. Okay, phosphoinopyruvate. Now there is something that is happening here which is very, very important. First of all, we know what is the rule. What is the enzyme that's going to catalyze this? Okay, we know that. Yes, we know that is what? Enolase. So enolase is the one that's going to catalyze this type of what? Reaction. So phosphoinopyruvate is going to be catalyzed by this enzyme enolase, but there is something that is happening here, okay? This one we're going to remove water. Okay, so we're going to remove water from this what molecule, which is the 2PG, which is phosphoglycerate, 2-phosphoglycerate. We're going to remove what? Water. So what are we saying? What's happening here? We're going to remove what? Water. So here we're saying we're removing water from this what? Reaction. If you're removing water from this reaction, what do we call that? We call it, what do we call that? Dehydration. Okay. So here dehydration is going to occur because we are removing water from what? 2PG by this enzyme inole, for you to have phosphoenol pyruvate. Okay, we're almost there. Then the final product, what do you think? It is what? The pyruvate. The pyruvate. What is now the pyruvate? So for you to have now this, the pyruvate, there has to be what? A certain enzyme here. We're going to use it. We are going to call it what? Pyruvate kinase. A very, very important what? Enzyme. And again, if you remember kinase, what do you think? There will be ATP production, either ATP usage or ATP production. But here under harvesting phase or payoff, the type of what? Phase is what? Energy production. So here we're going to produce what? Energy. So here we're going to put what? Like here we're going to put ATP, and we're going to have ATP. Okay, so we're going to have ATP production. Now, as you can see, how many ATP do we have? If this is 1, then we're going to have how many? 2. Okay, because there is also another reaction that is happening, okay, at the same time. So we're going to have what? 2 ATP, 2 ATP. Now, if you use mathematics now here, okay, so let's say we said we're having about what? 2 ATP, okay, and we are using up 2 ATP. So here we are using, so we use this one, and what did we gain? So we gain how many ATP here? We gain about what? 2 plus 2, we know that will be what? 4 ATP. So we're going to have 4 ATP, which we are going to gain. Now, since we use 2 ATP and we're getting 4 ATP, then what are you going to, for you to have the net ATP, what are you going to have to do? So net ATP, what are you going to do? You're just going to say 4 minus what? 2, for you to have what? 2 ATP. Okay, so you are going to have 2 ATP as the what? The result. If you come back here, what did we say? We're going to have 2 ATP because you're going to remove the one that you used from the what? The one you gained, okay? The one you gained, that was about 4. Just say 4 minus 2, okay? And that's where we end from, okay? And don't forget to subscribe and like the video, okay? All the best, stay well.